announcement. So I think perhaps we can start from questions, if any, and then we'll start from where we stopped. So, yes? Mm-hmm. We're gonna discuss that extensively. Okay, so I'll hold for that question. Another question? Mm-hmm. Yeah. Okay, that's a good question. I think that, I mean, the way I phrased it in the title is when it's easier, okay? Which doesn't mean that is the best choice. Right? It really depends, I mean, when it's best, it depends on the question you want to ask. Right? And obviously, studying what happens in a cell in balanced exponential growth, it's a very, very specific condition, okay? Which is very different from what happens to a cell that is entering a stationary phase, or a cell that is not growing or that is in a new environment, etc. And also, I mentioned this, but I want to mention again, it's not granted that, let's say, the exponential phase is the typical scenario where cells are, okay? Meaning in a even more strict sense for people that, let's say, have thought about this, it doesn't mean that what is evolutionary optimal, so what is selected, that basically bacteria that grow faster are always better, okay? And this is empirically, so I mean, it's not what we see. So if the exponential phase was, the most important phase was the typical phase bacteria are in, what we would expect is that the bacteria that there are only bacteria that grow very fast, right? Or all the bacteria should be able to grow fast. There are many bacteria that grow extremely slowly, painfully slowly. That takes like days to duplicate. While for, I mean, E. coli in fast exponential growth, lab-adapted E. coli in fast exponential growth, it can duplicate. One cell can become 2 cells in 20 minutes. Okay, so you have bacteria that duplicate in days. Okay, so this is to say that the exponential phase is a very specific choice. The point is, it's easier. So an analogy that I think it works is, if I want to study gravity, I want to study these things falling, where is the best scenario to study is in the vacuum, okay? Now, the vacuum is a condition that it's very hard to realize. So it's extremely hard to realize in the vacuum, and in our experiences, if I have a sheet of paper and this book and I let them fall, they will not arrive at the same time to the ground, right? But when we study gravity, this is the first thing we consider, which is something that is realized in a condition that is very far from our everyday experience. But somehow we have an understanding that that all the other things are complications. Right? So if you want to take this analogy, in some sense, exponential growth is physiology in the vacuum, okay? Which means that then if you have something, if you're observing something in outside, if you ask a question about the bacteria that are happily living on the surface of the sea, what happens in the vacuum, it's not really what tells you. It's the same way as if you have something sinking in water, what happens in the vacuum tells you very little, okay? So this is the idea, right? The idea is we first understand what's happening in exponential growth because it's easier. And then having understood this, we can understand what happens outside exponential growth better. Okay? If there is time, I can make an example of this. So I want to open this because I like to look outside. But I'm not able to. How do I do this? Here? Okay. Good. So I feel less. Okay. Very good. So did I answer your question? Okay. Other questions? Yes. So the equation we started is to say that if you want the mass of proteins in a cell, let's say call it proteins like this, it's something like this. Okay. This is not. This is in a sense is an assumption. It's something that makes sense. Okay, but it's not necessarily the most general things you could expect. So here what we are saying is, I mean, let's say I can always think that I have something like this, that there is a rate at which ribosome translate, and then there is a mass of amino acids, and the mass of protein is basically related to how fast proteins are built. This is true all the time. The point is that there is another step that in the notation that I introduced at the end, so we say we have conditions, right? We have some environmental conditions or experimental conditions. Right? And this experimental condition can be whether we put an antibiotics or not, in how much concentration, what are the nutrients, what is the temperature, so on and so forth. And somehow, what we said in the discussion so far is that, in principle, I could say, well, the mass of amino acids is independent of that. Amino acids are always amino acids. But in principle, for instance, this rate here could depend on E. Okay? And NR active could depend on E. Right? And in principle, this could be something where you have the total amount of ribosomes minus the inactive ribosomes. Okay? And in some sense, what we, in the first discussion we considered, when we were changing nutrients, we said if E corresponds to change nutrients, if, say, E changing nutrients, okay? What we say is that if we change nutrients, this K is constant. Okay? And also, this number here is constant. Okay? This is, in some sense, an assumption and an observation. Okay? Whether it's an assumption or observation, it's mostly how you want to tell the story, okay? Whether you want to say, oh, I thought about this hypothesis and I tested in data and it works, or I look at the data and I discover that this is true, but this is irrelevant. I mean, it's like just a chronological order. In principle, for instance, this K, you could imagine that depends on a lot of things. I mean, it could depend on the nutrients because this could depend on how many amino acids are available, right? So I'm saying there is a ribosome, right? And these ribosomes have to produce proteins, and amino acids have to arrive. In writing this, what I'm saying is the only thing that matters is how many ribosomes I have, right? So ribosomes are there, and somehow there is enough amino acids that the limiting step is the number of amino acids, right? So is the number of ribosomes, sorry. So the limiting step is the number of ribosomes, right? So here it's telling you, this equation is telling you that if I increase by 1 the number of ribosomes, implicitly at least, if I increase by 1 the number of ribosomes, I will increase this rate by 1. Okay? It's a bit more complicated than this, but this is the sort of hypothesis. So that somehow I can neglect how many amino acids there are. I can neglect the messenger RNA, right? I'm saying, in principle, if there is no messenger RNA, I will not translate anything. Here it doesn't enter, right? Sort of I'm saying that doesn't count. So there are, at the end of all of, the last thing I want to do is to give you a sense of what, how you can extend, and there are people that extended it to consider these other factors. Yes?
In one of the slides that you have shown yesterday, the R square was almost 1.
The
R square
okay.
was almost 1.
Mm-hmm.
So as if it's a deterministic.
Which plot?
I don't remember, but you
For instance, this one?
R square.
R square?
Yes.
This one. This one?
There was a schedule of R squares, probably another one.
I don't remember another one. This was the only one, but you see that this is like pretty, there is not much dispersion, okay? And the reason why there is not much dispersion is because, let's say, people that did experiments were very good, okay? And so this becomes very replicable. And because it's really a constraint, right? It's not a correlation. It's not something that statistically, it's really something that we can say that there are deviations, but these deviations are negligible. So for instance, all the fact that we didn't count, right? That, for instance, another aspect is that you need energy for this process, right? And you could think that energy limitation is very important, right? And different nutrients provide more or less energy. And so the ability of ribosomes to produce proteins could be very different depending on the energy availability, so depending on the nutrient. So you could expect that this depends a lot on the nutrients because there are nutrients which are more, which are a better source of energy and others are a worse source of energy. If that was the case, you will see a bigger dispersion. There is not. You could say, okay, perhaps here I see some deviation, and that deviation is due to that aspect, but that deviation is small, okay?
And there are only 4 experiments on each one.
There are?
4 experiments.
This is like more. So the color points are 6. On the other one, no, there are more. There is, 4 here, and 4 here, if you neglect the circle with the numbers. Okay? Yes? No, you don't change K. Right? K is a property, it's how fast a ribosome elongates. So how fast the ribosomes produce, so attach amino acids to the polypeptide. No, what I'm saying is if you increase NR by 1, let's say, not in a real, what you're saying, I think it's correct, but because you have in mind the pie chart, right? Where you increase the, yes, that's true. What I'm saying is if you take this equation and you are looking how many proteins you are producing, right, at a given instant in time, what you have is that if you increase NR by 1, you increase this rate. Right? Now the point, okay. Other questions? There was another question. No? Okay. Good. Okay. Okay. So I think we can start from where we stopped yesterday, which is more or less what is already written there, but let me rewrite it. So what we said is basically what we are doing now is to build a sort of simple theory where the main components of this theory is what is highlighted over there. So the idea is we have two processes that contributes to the growth. One is translation, and another one is nutrient metabolism, let's say. And this is performed by these two sectors. And so when we consider different conditions, what we have is we have that the growth rate in a given condition is equal to both these terms. So it's equal to KT, which could be condition dependent, phi r, which could be condition dependent, minus phi naught, equal to Kn, which could be condition dependent. Okay? And all of this is subject to the constraint P plus phi r equal to phi max. Okay? So this is the model we either starting from or arrived to. And now what I want to do now is to show you that this model reproduces what we observed. Okay? Before doing that, I want to take this model and, between quotes, solve it. I mean, it's a simple system of equation, but tells you what do we understand or well, actually, let's first see that this is consistent with what we saw in experiment. So how do we test this? So we have this equation, and now we can imagine to do an experiment where we change the environment, the experimental condition in some way. Okay? So for instance, we can say change E by changing nutrients. Okay? Now, if we change the environment by changing nutrients, this is a specific, you can imagine this as thermodynamics, right? So you have the experimental condition, okay? And you can change the experimental condition. You can change axes of the experimental condition in different ways, right? You can change volume, you can change temperature, okay? So you can think at this as you can think at that as an equation of state, and you can think of transformation as changing one axis in the possible value, okay? So for instance, what you can do is that you can change nutrient, okay? So if you change nutrients, there will be some parameters or some properties that are fixed, and others that vary, okay? So these, in principle, you can vary everything here, but if you change nutrients, this is equivalent to do a transformation with some parameter fixed. So what is the parameters that we have to fix if we change nutrient? No. It's one of the parameters appearing here, okay? You have all these terms. In principle, all these terms depend on what is the condition, okay? And I want to know if, and so this depends, for instance, for us, the conditions will be changing nutrients or adding antibiotics, okay? So if we change nutrient, not all of these parameters change, okay? So some parameters will be fixed. One parameter will be fixed. So which parameter will be fixed? If you change nutrients, do you expect the growth rate to vary or not? Yes. Okay. So if you change nutrients, you expect Kn to vary or not? Kn is how much so it determines the flux from nutrients to amino acids. So it tells you for each protein that I allocate to the metabolism of nutrients, I produce amino acids with this rate. Do you think that that depends on the nutrient or not? This is Kn. Yes, right? So it's different. So Kn changes. Do you think that Kt depends on that? In this simple model, no. Because if I change nutrients, I change this rate Kn, okay? But once I produce amino acids, then I change the rate at which I produce protein, right? So it's unaffected. So change E by changing nutrients, it's equivalent to fix Kt, okay? So in my, let's say, equation of state, what I have to do is to fix Kt. So if I fix Kt, what I discover is that this is fixed, okay? So all these relations are always true, but since this is fixed, it's convenient to write this one, right? So since this is fixed, what I discover is that lambda of E is equal to Kt phi r of E minus phi naught, okay? This is true, okay? And using this second equation, I can also write Kt phi r is equal to phi max minus phi p. So this is equal to phi max minus phi naught minus phi p of E. So what I discover is that if I change nutrient, what I expect is that phi r of E, right, from the first one, it's equal to phi naught plus lambda of E over Kt.
To make sure, Kt is the rate of amino acids being imported.
Kt is the rate at which mass is added, at which mass from translation is added per unit of ribosomes, basically. Okay. So there are convention factors, but it's basically how fast ribosomes produce proteins. Okay. So it's telling you if I take one ribosome, how fast I produce proteins from amino acids. Okay. And then I can take the second equation, okay? And from the second equation, I have that phi p of E is equal to what? Is equal to phi max minus, did I do something wrong? No, it's correct. Phi max minus phi naught minus lambda over Kt, okay? Lambda of E, okay? So this is like, if you want, are transformations at fixed kappa t. Let's call it kappa t translational capacity, how fast you translate. So these are transformations at fixed translational capacity. Okay? Then I can say, I can do something else. And I can say, now I can change the experimental condition by adding antibiotics. Okay? So this is equivalent to fix what? Well, no, not really. What you fix is if you add antibiotics, you are messing with phi r. Right? So if you add antibiotics, what you do is that you are, no, my cursor doesn't work. So you are messing with the second part, right? Because you make, so the type, I mean, this is not true for all types of antibiotics, but for the type of antibiotics that we care about, you are messing with the second part, okay? With how fast you produce proteins out of amino acids, okay? So if you change by adding antibiotics, what do you fix? Kn. You fix, that part is untouched, okay? And so this is equivalent to say I fix Kn, okay? So if you fix Kn, what you get is now it's convenient. I mean, this is, the point is now this equation, this changes, this changes, this changes. This is still true, but it's not obvious to find a clear relation. I mean, it's simple math, you can find it, but it's better to work with this, okay? So if you work with this, if you fix Kn, you get lambda of E equal to kappa n Vp of E. And then I can use the second equation to write this as phi max minus phi r of E, from which I get phi p equal to lambda over kappa n, and phi r equal to phi max minus lambda E kappa n. Okay? Yes.
I'm just got confused with the thing that when we mutated this cell, then we were changing this lambda rate. And also like here, when you're adding antibiotics, you are playing with Kn, which can, in terms will sort of change that rate. So how these two are different?
So the idea is, if you do something, if you change the experimental condition, okay, in principle you are changing everything here. You are changing the growth rate, you are changing how many ribosomes you have, you are changing how fast ribosomes work, you are changing how fast nutrients become amino acids, okay? You are changing everything except the things that do not depend on E, okay? So this is the thing. Now, if you want to test this, there are two smart ways to do it, right? Given the form of this equation, one smart way to do it is to do an experiment where you change the experimental condition such that this is fixed, and another set of experiments where you change the experimental condition such that this is fixed, okay? I mean, in principle, you could put on the same plot everything, right? But in order to understand what's happening, it's better to reason in transformation or change of experimental condition that fix this or that, okay? And you get these two. And now, this is exactly what we observed, okay? So this is exactly what we observed. So this part here is exactly what you observe over there, okay? What we observe over there, it's this relation here, so that the fraction of ribosomes increase linearly with growth rate, and that Kt is related to translation speed. And then this is again this part here. If, sorry, it's not completely this part, but it's both of the prediction. So what you see here is that if you change nutrient quality, so you move on this line, okay? So this is the first type of transformation. What you see is that you are moving for phi r in this way, okay? Which is this relation. And then this relation here, it's represented by this line here, okay? That you can show as the same slope and value, okay? If instead you add antibiotics, what you're doing is that you are changing the growth rate, you are changing the sectors, fixing Kn, okay? So if you do that, what you do is that for phi p, you have line with this slope, okay? Which is exactly what you have, okay? So where the slope depends on the nutrient, right? Because it depends on Kn. And for phi r, you get, well, phi max minus this term here. You get that you move this way. Okay? Good. So what else can we do with these to further test this? Okay? Well, first of all, so is this clear? So why the model reproduce what we observed? Okay? So this is a good, now we have a good framework to play with, and then we're gonna play with this. So let's say another things that you can do to test it. But before doing that, I mean, here you have, let's say, we want to express, we have, I mean, in general, I mean, we can express, the idea is that this Kn, Kn, and Kt are properties of the experimental condition. So are the input we put, right? So the way to interpret, to look at this is that Kn and Kt are inputs. Okay, so are the experimental choice. If we want to do an experiment, what we do by choosing the condition is choosing Kn and Kt, and lambda, phi r, and phi p are the outputs. Okay? So what we can find is that we can express everything as a function of Kn and Kt. Okay? So if we take that equation and we can write lambda of E is equal to Kt phi r of E minus, well, I'll drop the E because it's like a bit redundant, is equal to Kt phi r minus phi naught, but is also equal to the second equation, Kn, which I can write using the third one as phi max minus phi r. Right? So then I can take these two equations together, and what I get is that phi r of Kt plus kappa n is equal to kappa n phi max plus kappa t phi naught. Okay? And so what we get is that we get that phi r it's equal to kappa n phi max plus kappa t phi naught over kappa n plus kappa t. Okay? Now, what does this equation tells us? So this equation, we can look at what happens to phi r. Okay? So we have R, we have P, and we have the rest. Okay? And so this rest here, well, this sum here, it's phi max. So what happens if we go to very large Kn? Okay? So if we go to very large Kn, kappa n, you see that these two terms becomes dominant. And you see that if you go to very large kappa n, so for, let's say, I can erase this part perhaps. So if you have kappa n much larger than kappa t, okay, what you get is phi r is approximately phi max, and phi p is approximately 0. Okay? So this means that we are in a situation where we have r and p is basically 0. Okay? Vice versa, if we are in a situation where kappa t is much larger than kappa nutrient, what we have is that phi r we see here, if kappa n dominates, if kappa t dominates, phi r is about phi 0, okay? Which is only inactive ribosomes, okay? So phi r, it's phi naught. And so basically you have, this is R, and you have mostly P, okay? Okay? Now, we can add to this picture what happens to the growth rate, okay? So we know that so the growth rate is equal to, what is more convenient to write? Yes? No. Okay, I'll comment this and then go to the growth rate. No, so this is, well, I'll do the growth rate and then I'll comment on this question. But this is what you expect. This makes sense. I'll explain you why in a second. So what you get here for the growth rate is that if you insert this equation here over there, what you should get is right, okay, yes. You can put this over there and you get K n kappa n kappa t over kappa n plus kappa t. Okay? Because you, yes. Good. So this is the equation you get for the growth rate. And so in this case, what do you get? In this case, you get that the growth rate is when kappa n is much larger than kappa t, you get phi max minus phi naught kappa t. And here the growth rate is phi max minus phi naught kappa n. Okay? So what is this telling you? So this is telling you, first of all, that you have this process here, okay? And the growth rate, it's dominated by the slower process, okay? So you have these two rates, how much one unit of protein that you allocate to the first process or the second process contributes to growth, okay? The slowest process, so in this case kappa t, and in this case kappa n, is what determines the growth rate, okay? So basically, it's like you imagine you have two fluxes, okay? And the slower flux is what determines what is the total flux, okay? So this means that if you are in a very good nutrient, let's say, for instance, what determines your growth is how fast are ribosomes. Okay? And what is the interpretation of this? If you are in a very good nutrient, okay, basically the first process, you don't need to allocate many proteins to the first process because the first process is already fast. Okay? So you don't need to allocate many proteins. I mean, remember that you have always this condition, right? So the rate of the first process, it's equal to the rate of the second process, right? But what happens is that if this kappa n is very small, okay, basically what you do is that you have to allocate, sorry, if kappa n is very large, you have to allocate many ribosomes to keep up with all the amino acids you are producing, right? So basically to balance these fluxes, you have to allocate many ribosomes, because what limits your growth is not the nutrient quality, but it's how fast you translate amino acids. In this case, which let's say you are in a bad nutrient, what limits your growth, it's not translation, is how much amino acids, how much substrate you are able to produce to then produce proteins. So in that case, you are in a bad nutrient, you have to allocate many resources to the first process, okay? So imagine that you are like, that we have to do a job together, okay? And there is someone that has to go outside, fish some fishes, and then there is someone else that needs to clean them, okay? If you are in a situation, and we want this process to be continuous and not to have backlogs, not to have accumulation, to be as fast as possible, okay? Suppose that we are in a situation where fishing fish outside, it's something that is very slow, right? It's a very painful process. Then what we are gonna do is so we are in this situation here, okay? So first getting food, and second one cooking and cleaning and cooking them. So if we are in this situation, we want to have many more people outside, P, than people inside cooking, right? Because the time it takes to cook one fish and clean it, it's much faster than the time it takes to fish it, okay? If instead we are in this situation, right, we can have one single person outside continuously keeping and throwing fishes to us, and then everyone has to keep up cleaning to process all the fishes that and cooking all the fishes that have been fished, okay? So this is the idea. Is this clear? Does it clarify the intuition? Okay. Yes.
Question about like analogies. Let me say like, can we consider the first case like, suppose you are taking like lactose. In the first case, you are like giving lactose in form of directly concentrated, not like form of lactose, but in form of the other sugars. And in the second case, you are giving lactose in form of lactose.
Yeah, I mean, you can think about two different nutrients. So for instance, here you have, I don't know, you give glucose plus amino acids. So you give a very good carbon source and amino acids already, so which is something that they takes time to produce. So it's something, it's some amino, so basically you speed up growth, right? So in that case, what limits your ability to grow? It's really the first one. In the second case, you give like cellulose. I don't know if this goes on. So it's like something that you really need to degrade, to break down. So you really need to allocate many internal resources to do the first step, okay? In this case, it's not much about I mean, you can also play with concentration. It's a different story. But let's say play with concentration with nutrients. I mean, I don't know if I want to open this parenthesis, but basically, the concentration with nutrients, if you give too much, some nutrients become toxic. Then there is a range where a very wide range where they grow, and the growth doesn't depend on how much you give them. The things that depends is when they saturate, but not how fast they grow. And then if you give too little, then it becomes important, but then it's like very sensitive, and you are at concentrations where then you immediately feel that you are depleting them. So experimentally, you typically are in a spot where there are enough resources, but then the concentration doesn't vary your growth.
This is not model in our model. If we add a lot of nutrients, it will become.
This is not how much nutrient, is what type of nutrient. Okay? So here, it's quality of nutrient if you want. So here, it's like this part here, right? This kind of experiment. What you do here is that you change nutrient. So here you grow them in a simple salt plus glycerol, simple salt plus glucose. glycerol plus casamino acids, glucose plus casamino acids, and then glycerol plus a rich medium, which is something with many things, and glucose with something with many things. Okay? So it's the type of, it's not the amount. The amount for us, it's not relevant for this discussion because determines if you give more food, simply you will have more bacteria at the end, but they will grow at the same speed. What we care about is speed, not how much bacteria you have at the end. Yes?
And you can increase the quality like infinitely if you
No,
use
this tells you you cannot increase the quality infinitely. You have actually a maximum amount of growth, which is this one, right? So in some sense, the max of your growth that you can achieve by changing nutrients is phi max minus phi naught kappa t. So this is lambda max. And if you, by the way, I mean this, okay. If you put like the values you infer for this, which is about 0.55, and you put the value of kappa t that you get from this value, you get the a growth rate that corresponds to an application every 15 minutes, which is very consistent with what you see in E. coli. Okay? So the idea is there is a limit of how fast you can grow, and that limit is determined by how fast ribosomes can elongate.
I mean, the kappa T doesn't have a constraint in our model.
Sorry?
PT doesn't have constraint in our model. Like it is constrained in reality or an experiment?
It's constrained in this model it's a parameter, right? So in reality it depends on the condition, and in reality it has a maximum. I mean, you cannot have ribosomes going at the speed of light, right? So the speed of light is not the limit, right? So you really have physical limit of how fast you can go. This, for instance, this parameter here, it's very sensitive to temperature. And so it's what determines the fact that, it's a big component of the fact that there is an optimal temperature for growth. But let's say there are limits, and these limits is a property of the ribosomes. Okay? So it's not, we are sort of, in my opinion, this is very cool because it sort of split the problem in two parts also. It splits the problem of what are the property of the ribosomes, and this is something that I can take out of a living cell and study by itself, in principle. Okay, so it's a problem of the machine, from what a cell is doing in different conditions. Right? So what you're saying is, now we are, let's say, doing the example of fishes and cleaning them. The people cleaning them can be more or less skilled. Right? If you are a cell, you are stuck with the people you have, whether they are more skilled or less skilled, right? Or the type of knife you use, right? The tools you have. You are stuck with that. The point is, you are going to do different things depending on the condition, but the tools you have are the tools you have. Throwing another things on the thing is, now you can say, this tells me what I can do with the tools I have. So that can also be used to think about, suppose that I want to change my tools, so evolution, what are the constraints to evolution? Okay? But this is another part. Yes?
Yes, sorry.
Sorry, yeah.
If,
Okay. No, go ahead.
so if you don't satisfy the condition, is any implication in the experiment?
This one?
Yes.
Yes, you will not see, if that condition was not true, you will not see this as a lie.
No, but in the experiment, like the coding may be sick, or
The?
the organism behind the experiment is sick, for example, or any other thing.
Well, the point is, when you, let's say, I interpret your question in a different way. So I could interpret your question in two different ways. So one way to interpret this question is, can this model fail? And the answer is yes. You could have, it's not obvious that this works. You can, in principle, if before changing this plot, I was telling you, or telling someone with a training in biology and they didn't know this plot, how does I mean, let's say if you train in molecular biology, so you have in mind the picture I showed you about the messy network, and I had to tell you, what do you expect from this experiment? Probably a cloud of point, perhaps with some correlation, but what you would expect is a cloud of point. Because that metabolic network is so complex that you don't expect any generality emerging, okay? So you are biased because you didn't study biology, or didn't study biology much, or most of you didn't study biology much. But the point is, what you expect is that this does not hold, so it's non-trivial. This is one thing. You don't expect these relations to be there. The second point is, the fact that we see lines, nice lines, is because we are changing the environment in a very specific way. So when you look at this plot, in this plot, you can see lines, right? If you put the lines, right? If you know that here you are changing something, and here you are changing something else. But if I remove the lines, I remove the colors, what you see there, it's a cloud of points, okay? So there is also something about how you do experiments, right? So when you do, in this case, this is successful, so also because you do experiments in the right way. You change one thing at a time, okay? I hope this answered the question. Yes, I think they fix it, but the point is, so since you are bringing it up, I think you will see in the lecture by Amir Eretz, but the point is, what you expect is that the growth rate depends on the concentration of nutrients, okay? And so how much nutrients you put. And typically, it has a dependency like this, okay? Now, this scale here, it's very, very small. Depends on the nutrients, but it's typically very, very small. Okay? So if you are above this scale, the growth rate is constant with concentration. Okay? And now, if you look at your culture, right? So you look at your bacteria growing as a function of time, how much nutrients you put, so basically, how much nutrients you put. So let's say that you are let me go over here. So we say, do we have the growth rate as a function of C? And these are something like this. Okay, see? Okay? So what you have is, if you look at the log biomass as a function of time, okay? And imagine that you do an experiment where you start here, 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 or here. And this is the initial concentration, okay? What happens is that if the initial concentration is much larger than this Kc, what you see is that this grows exponentially to different values. Okay? Then if C0 is about Kc, then your goes lower also here, right? The point is, we care about this. Okay? So there is a wide range of initial concentration where this does not apply. Okay? Is this clear? Yes, the concentration is always here. So being always here, it doesn't matter. I mean, you can test this, but yeah. Okay. I mean, this is the first order answer. There is a second order answer that actually, if you put a concentration which is too high, then you start to accumulate toxins, and so then you have other problems. So, but it's something microbiologists know very well, how to do with this. So, but there is a range where let's say growth is limited by nutrients and you are exponential, etc. But the point is, I mean, the key part here is whatever you are doing here, what you do is that you guarantee, you try to do it such that you are in exponential growth. Okay? Yes? Yes. You solve, so basically, I mean, you have 1, 2, 3 variables, okay? And 1, 2, 3 equations. So you can see it, so if you want, we can solve it. So you have, from that equation, you see that phi r, it's equal to both phi naught plus lambda over kt, right? And you have that the phi r is also equal, from the second equation, to phi max minus lambda over kn, okay? Now, when I'm writing this, in principle, everything depends on the environment, on the experimental condition. And so if we equate these two, what you get is lambda 1 over kn plus 1 over kt. It's equal to phi max minus phi naught, okay? From which I get lambda is equal to phi max minus phi naught. 1 plus kn plus 1 over kappa t to the minus 1, which is exactly what I wrote there. Okay? Good. Other questions? Okay. Yes. I'll show you a plot. So, yes.
So, just looking at, can you say that in this graph we are in bad nutrient condition?
It depends where you are. So this is a relatively fast growth. This is fast growth, actually. So all of these actually, if you ask microbiology, all of these are very good nutrient conditions.
But just because if we are in very good nutrient, then there we are changing nutrient, right?
Yes.
So KT should be fixed.
KT is fixed.
So lambda, I can.
Yeah.
Like,
You can infer what is Kn, what is kappa n from here. So in this case, we are on the line changing nutrients. So for that plot, you have that phi r is equal to, So you can look at this equation, right? So for that plot, what you're doing is that you are fixing lambda, you're changing lambda by changing kappa n. So
at some point I'll be in good nutrient.
Exactly. At a certain point, saturated. Yes. So if you there is one. So basically, what you have is that as a function of kappa n, you have this equation here. So what you have is that lambda, if you fix kappa t as a function of kappa n, does this, right? We don't see the saturation because we are not plotting against kappa n, right? So each of these points corresponds to a different kappa n. Okay. But also in this, for kappa t is different, but for kappa n, in some sense, we don't have a way to measure it that is not looking at the value here or fitting this line. So fitting this line here, right? So kappa n is the slope of this. Okay? Good. Okay, so we have this now. What we can try to do, so the next things we want to do is to say, okay, so so far we have a model that reproduces many experiments quantitatively, everything works. We can try to do another prediction, okay, of something that we didn't really never put in. Okay? And so, yes? No, it's different. No, it's different. So in this case, it's whatever this kappa t is. In this case, it's this number here. Now, if you are changing, if you move, so let's say that you move on these axes by changing kappa n. Okay, so you fix kappa t as you usually do. I mean, if you don't add the antibiotics. So you are basically, suppose that you are on this line here. Okay. The growth rate for, kappa t is fixed. Okay. So the only thing you can change is kappa n. So in this case, you are at the maximum growth rate. In this case, you are at very slow growth. Okay. This is what we have at the beginning. Yes, yeah. It's just, it's exactly the same. It's just writing the solution not in terms of lambda, but in terms of these parameters. Right? So when you write this equation, right, what I can do is to write phi r, for instance, as a function of lambda and kt. Or I can write phi r as a function of lambda, I can write phi r as a function of lambda and kt. Or phi r as a function of kt and kn. Or phi r as a function of lambda or kn. Right? I can do all the combination. These are always, all these, the equations that I will write, they are always true. The point is, when I do an experiment and I change some parameters, in order to see what I expect, it's better to write the condition where the parameter is fixed. Right? I can make a prediction choosing the right variable. Okay? Good. So I want to derive another prediction. And the other prediction, let's start from this equation here. So what we see is that the growth rate, so let me rewrite it here. So at the end, what we see is that the growth rate depends on a few quantities, right? Depends on phi max minus phi naught, and then kn, kt, kn plus kt. Right? So we extensively tested and used the results where we change kn and kt. Okay? So another thing that you can do is here you have another parameter. Right? Well, you have other two. Phi naught, it's messy, but you have phi max. What is phi max? Phi max is, in our pie chart, is how big is this one. Okay? So phi max is 1 minus phi q. Okay? And now this sector is untouched so far. So one thing you could do is to say, what if I perturb this q? How do I perturb this q? For instance, I make the cell express a protein that they don't need, that doesn't do anything, but always express at a fixed value. Okay? So what you can do is to say, now what I do is that I have R, I have P, and now I have some protein, which I call U, which I have phi U, which is a fraction of unnecessary proteins. Okay? Okay? So I basically make a construct inside the cell such that they always express this protein, and I can have, there are ways in which you can choose how much of these proteins they express. So you can control how big is this phi U. Okay? Is this clear? The, between many quotes, experimental setting? Okay? So you have a tool where you can say, now I choose, for a given experiment, I choose how big is this phi U. And you can do it in multiple ways. Yes? No, it's a so it's increasing Q. It's not a subfraction of P. Now what I'm doing is that a cell, what it does when it grows is that it changes R and P. Okay. Now, before I had this constraint, so I'm changing this constraint. I'm saying now phi P and phi R don't sum to phi max, but they have to sum to phi max plus phi U, minus phi U. Okay? Right? So I introduce this. So now the constraint is phi P plus phi R is equal to phi max minus phi U. Right? Because it's phi Q plus phi U. So basically, I'm increasing artificially how big is phi Q. Okay? Is it clear, the setting? Before I move on, I want the setting to be clear. Okay? So you say, okay, now you cannot go up to this point. You have these proteins you express. And this protein doesn't do anything. It just stays there. The cells keep producing it, but doesn't do, yeah.
It's not clear for me that why you extract U from Q.
Because I'm adding it. So this Q is like all the proteins that are here. So it's not, sorry, I see because of the, so these should be the same. And now, sorry, so this is exactly the same as before, but now I have U. Okay. Is this better? So the part of Q is exactly the same as before. What I did is that I added a slice to Q. Okay? And I decide how big is this slice.
Isn't it just renaming? Because
It's not renaming. It's really, I mean, all the things that are in, so Q is a sector, so it's all the proteins that are there to do some functions, that is always there, doesn't depend on the growth condition. Okay? Now, and this is fixed. So you change growth condition, those proteins are always expressed at the same level. What we are doing now is to say, now in addition to this, you also express these other proteins. Okay? At a fixed level that we decide. Okay? So we have an experiment where we decide how big is this phi U.
so phi max is constant.
No, phi max decreased, right? Because now phi P plus, so phi max is 1 minus phi Q before, okay? Now phi Q, sorry, now phi max is 1 minus phi Q minus phi U, okay? So this is, you can imagine Q as an incompressible part, right? You can always change P and R, right? But you cannot change their sum. Okay? What we do is we are considering a way to change their sum, right? Where we fix the sum. So now the sum is not phi max. Now the sum is phi max minus phi U. Okay?
So is the sum of P and R is phi max minus P that phi max must be again 1 minus phi Q.
Phi max is still 1 minus, yeah, I mean, yes. Phi max is 1 minus phi Q, but then you have also this, yes. So, yeah, I mean, it depends how, yeah, phi max is the one of before, but now we have this extra term, okay? Is this clear? Is this clear?
You're adding a parameter to like change the maximum value we can.
Yes, exactly, yes. Exactly. We are adding a parameter where you can change the maximum value. So basically you can repeat the calculation, and what, I mean, if we start from here, right, we have the same everything, except now we can think that this phi max becomes phi max minus phi U, right? So effectively what we are doing is that we are sending phi max to be phi max minus phi U. Okay? Good. So what we have is that now lambda is equal to phi max minus phi U minus phi naught. Okay, so I can write this as lambda is a function of phi U, Kn kappa t, kappa n plus kappa t, which I can write as phi max minus phi naught times 1 minus phi u over phi max minus phi naught, kappa n kappa t, kappa n plus kappa t. And this is equal to this term times this term is equal to lambda when phi u is equal to 0, 1 minus phi u over phi max minus phi naught. Okay? So what I did is to write, this is what we predict, and then I rewrote our prediction where I wrote the growth rate as a function when we have this expression of this unnecessary protein, as a function of the growth rate when the unnecessary proteins was not expressed, okay, times the effect. Okay? And sort of the prediction is that when we add these unnecessary proteins, we decrease the growth rate. Okay? So if we look at the growth rate with phi u, we have, as a function of phi u, we have 0. When this is 0, we have what we had before. And then these decrease linearly with some slope that we can predict. Okay? And it works. Okay? So these are the experiment that we did. So this IPTG induction is just the way you control the level of the expression. So here it's the percentage of the total protein, so really what is phi u. And then these are three different values, three different nutrient conditions, which corresponds to different values here. Okay? And what you predict here, if you want, is a simple way to see it is you start from here and then you have to cross to go to 0 at the same value of phi u, which is equal to phi max minus phi 0, which is exactly what you get here. Okay? Yes?
It may be irrelevant, but in this setting, it was first to reduce theoretically, then the verification was done experimentally.
Yeah, you can imagine that. The chronology. No, this is in the same paper, but the point is let's say it's hard to know. It's a let's say there is a way in which people build a story, and then there is a way in which people tell the story, right? This is also true if you write a book, right? The way you write a book is not the way the book is read, right? So I don't know. The point is, it's clear that this is something that you don't need, let's say.
I'm just curious.
Yeah. It's an evidence for the model or the framework or the theory that it's not something I would say we put in, right? It's not, let's say, it's a prediction that we really derived, right? Good. Yes? I think it's like, it's the typical thing we do in physics, right? So you write a model, right, that makes some prediction, and then you say, okay, let's take this model seriously. What other prediction can I do with my model? We did this, and you test it, right? And you say, okay, now I can see if the model fails or not. No, it's to test the model. Right? It's to test it's really like, I think epistemologically, right? You have models where you have parameters, right? And these are not fundamental parameters. It's properties of parameters in the sense it's things you can manipulate, right? And we manipulated some, and if you have other parameters, you can try. You want to try to manipulate all of them. So one parameter we didn't manipulate is this phi max, and this is a way to manipulate it. Right? And in some sense, the fact that it works is telling you, okay, it's consistent really with the interpretation that we give. Okay. Other questions? Yes.
Why are you calling it overexpressing? Why are you doing overexpressing?
Overexpressing because, okay, this is in some sense jargon, because this enzyme here, it's something that the cell express it, or could express it. So it's already in this picture. What we are doing is we are expressing it even more, right? And we tune how much more is expressed. Okay? In principle, you could do it with the other proteins that are not part of it, and then it's, yeah. Okay? So what you can ask is this a property of E. coli or it's more general? You can do this experiment with other bugs, and this is like different bacteria, and what you see is that it applies, that you still see lines when you plot RNA over proteins as a function of the growth rate. Now, what you see here is that the slope here, which is the translational capacity, it's different, okay? So the slope is typically higher, which means that the translational capacity is smaller. So KT is more, kappa T is smaller. Okay, they go slower. This is also interesting because it's a eukaryote. People did it also in yeast. So this seems to hold. Okay, doesn't need that is a universal rule of life. There might be organisms for which it doesn't work, but at least there is a non-negligible class of organisms that behave like this. Okay? Good. Questions? Yes.
A small one. What would happen if we increase the production of the necessary protein such that the whole of the RRP species covered? Like, what would happen?
There is a point where you grow it so much, right? So if you grow it enough, then the cells don't grow, right? There is a point, you see also in the plot. So basically, if you make these to occupy all the space, okay, then there is no possibility for cells to grow, right? And this is the point where the growth rate becomes equal to 0. Okay. Now, experimentally, it's hard to go to very small growth rates, but yeah. Other questions? Okay, so I have 10 minutes, and in these 10 minutes, I can show you how this, very quickly, how this can be extended. Then I tell you what we do tomorrow. Okay, so one thing is that this is, one reason why this all works is because we only make steady state considerations, so we put ourselves in balanced exponential growth. But the point is that there is an whole story about how this is regulated, right? So suppose that you are in two different conditions. Then if you change condition, you change nutrient, you have to change how much ribosomes you produce, for instance. And the cell has to do it, right? And there is a feedback mechanism, which is governed by a molecule called PPGPP, that basically controls this feedback, right? So there is a sensing that a cell does about the internal state to regulate how many ribosomes it has, okay? And so one can sort of try to build a out of steady state description of how a cell changes these sectors, okay? So one can use what you know about the fixed point to move and write the dynamical system.
Excuse me.
Yeah.
The cell regulates the number of ribosomes just by producing the new ribosomes or just by activating the inactive ribosome?
It's a very interesting story, both. So what this PPGPP does is that it both increase the production of ribosomes. It also changes the activity of ribosomes, making them active or inactive. Yeah, both of them, yes. As usual in biology. Okay. Okay, so another way you can extend this is you have a simple process. Now forget about the lines here, the regulations, and you can extend it by adding steps, okay? And I will not go through the details, but I'll just give a flavor of this. So I don't expect that you follow everything now, but I want to give you a flavor of how you can extend it. So one way you can extend it is that you can say, now I take this step, okay, how you go from nutrients to amino acids, and I split it into two parts, which I'll call with some end-waving way, catabolism and anabolism, which is not really true. But the idea is you have a first part where you sort of are extracting energy from the nutrients, and you build and you have byproducts, which are called precursors. And then you have a second step where you build amino acids out of these precursors. Okay? And the idea is you can do experiments where you have, I mean, you always need multiple sources of nutrients, but you do experiments where you have an influx of carbon, and the influx of carbon is what you need to do this first step. Okay? And then you have an influx of nitrogen or phosphorus or sulfur, so another nutrients that you are changing. And by changing that, you are changing this step here. Okay? This is the idea of the experiment. Plus, you have this one. So basically, before you were manipulating nutrients by changing the type of carbon source, typically. I mean, you are also adding amino acids, but you are changing the type of nutrient in general. Now you say, instead of changing the type of nutrients, I have two things I can change. I can change the carbon source, what, for instance, sugar I give, glucose, lactose, etc. And I can change the nitrogen source. Okay? Or I can change how effectively the cell metabolizes glucose or lactose. Okay? So I can mess up with the enzymes also. So I can, okay, so this is the idea. In terms of models, which probably for you are easier to understand, hopefully, the idea is that now we go from this and we split these in two parts. So now our pie chart is Q, R, C, and A. Okay? And now what we have is something very similar from before. We have Kc, Vc, Ka, Va, Kt, Vr minus V naught. And then we have Va plus Vc plus Vr equal to Vmax. Okay? Same setting. We are just adding one step. So we are opening this box. The idea is that now we are going to do experiment where we can change Kc, or we can change Ka, or we can change Kt. Okay? Now you know how to derive predictions, right? How do you derive predictions? Well, if you change C, okay, you do this transformation, you change C. So you limit, let's say, you limit by carbon source, you change the carbon source. Then what you're doing is that you fix Ka, fix Kt. So this means that in this condition, Phi equal to lambda over Ka, Phr is equal to Phi0 plus lambda over Kt. Okay? And then using these two equations, you can derive what is Phi C will be equal to Phi max minus Phi naught minus lambda 1 over Kt plus 1 over Ka. Okay? Same math, same simple math as before. Now you can say, I change a lim. I change A, I change the nitrogen source. So then it means that you fix Kc and Kr. And you can derive similar set of predictions. Okay? And so in this case, you will have, well, it's simple. You will have phi c equal to lambda over Kc. Phi r will be the same. And phi a will be phi max minus phi naught minus lambda. Well, okay. So in the math, it's very, now you see the regularity. The math is very simple, right? When you change something, so you change one of these capacities, let's say, you change kappa something, you have a corresponding sector, a corresponding set of proteins. All the other ones are linear in the growth rate. So if you change C, A and R are linear in the growth rate, and the other one is the maximum minus all the rest. Okay? So the proteins that corresponds to one transformations are the ones that are decreasing with the growth rate when you change that parameter. Okay? Another way to interpret it is, if I have many processes, right, I have people fishing, I have people bringing the fish here, I have people cutting, I have people cooking. If I mess up with a tool, okay, as I mess up with a tool, what happens is that I have to reduce, if I reduce the number of baskets that I have to bring people here, what I have to do is that I have to increase the number of people that are using the baskets and decrease all the rest. Okay? This is what's happening, right? So you decrease all the rest, it means that as you make the system more inefficient, you reduce the number of people in all the rest, except you increase that. I hope it clarifies. So what you get is, for instance, RNA protein, if you do, NC is the same as A. So if you do both type of transformation, right, you are both here or here, depending on whether you have empty symbols or filled symbols, you get that phi r behaves exactly the same, whether you change the nitrogen source or you change the nitrogen quality or you change the carbon quality. Here it's a proxy of phi c. So what you do is that you look at what happens to this phi c, right? If you change carbon source, you are here. If you change nitrogen source, you are here, as you predicted. You can do the same for phi a. If you change the carbon source, you are here. If you change the nitrogen source, you are here. Okay? You can limit, you can change kappa t, and you get what you expect. So phi r increase like this if you change r, and if you change c as a like this, and then the other two are linear as expected. Okay? So this is to say that we started from something simple. Now the same sort of principle apply also to a new set of experiments, right? And from the model perspective, I mean, the beauty of this is that from the model perspective, it's a simple addition. I mean, it's something that I rushed through in literally 5 minutes. From the experimental setting, it's it's a completely new set of experiments, completely new kind of conditions, completely new kind of observables. So you have to look at some proteins which for which you can infer what is the size of these. So very simple prediction in a setting where you have many choices, many complications, many details. Okay? Good. I'll stop here. So what I want to do tomorrow, and then tomorrow we'll decide whether to do also Monday. So what I do tomorrow is to show you an example of how you can further apply this in the context of antibiotics response. So basically, you have many types of antibiotics, and depending on how much antibiotics you give, cells grow differently. And I'll show you how you can use these to predict what happens. Okay? Quantitative breaking. Good. See you tomorrow.
Why do we have so much linearity here? I mean, like, it's a biology process. Nothing should be linear. Why do we see?
Because, I mean,